Hello and welcome to our Ecology and Evolution series seminars in our series on environmental diversity. This is our second in the spring series, and for those watching this recording, we had some technical difficulties with the live version, so we're lucky enough to have our two speakers, Vinuri and Sophie, come back online again to record this session. So there are no questions here, but we have the two presentations. So our seminars aim to share the world-leading research and engage our environmental and evolutionary community together, and it's brought to you by the University of Adelaide. And every month we have speakers from a range of topics. And today we're lucky enough to have two PhD students presenting their CCSP presentations. That's at their 6-month mark at the start of the PhD. And first up we have Vinuri, looking at the diversity of marine microbes that colonize on marine plastics. So without further ado, I'll hand over to you, Vinuri.
Thank you, Bowie. Okay, thank you all for watching. My CCSP, my PhD is going to be on the hidden inhabitants of marine plastic. I'm Vinuri and I'm supervised by Bronwyn, Gillanders, Vilma Perez, as well as Linda Umbridge. To begin with, so this graph basically shows the global plastic production by industry in the year 2015. On the x-axis, you can see the years starting from 1950 until 2015, and these different colors represent the different industries, and gray includes other, but that includes healthcare and agriculture as well. However, these bars basically illustrate the average number of years the plastics are used within this industry before they are discarded. So if you look at the other industries, we've got building and construction, as well as textiles, producing a lot of plastic, 72 million for building and construction, as well as 65 million for textiles. And what's interesting to see is how the average shelf life of these plastics dramatically decreasing. And the pink color, the industry that's represented by the pink color is packaging, which is the greatest production of plastic, which is 161 million, and the average time that plastic is used within this industry is less than 6 months, before it is discarded, which is not a long time. However, what happens to this plastic? In 2015, this study produced that the plastic waste generated was 6,300 metric tons, out of which 9% was recycled, 12% was incinerated, and finally 79% was sent off to landfill or accumulated in the environment. And by accumulated in the environment, it can be either on land or in the ocean as well. If you look closely at the waste that's been discarded in the ocean, around 60 to 80% of that waste is going to be made up of plastic. And it's not as straightforward as you think to call all of these plastic as one thing. What I mean is, here we've got multiple different types of plastic items, daily items we've used. And if you've got a plastic water bottle on your table, if you look at the underside of it, there's potentially a triangle with a number enclosed. And what that basically means is what type of plastic your plastic water bottle is made up of. This is what I meant by plastic. You can't generalize all of this as one thing, as all these multiple plastic items are made up of different polymers and additives in order to cater to various needs. However, what happens to this plastic after they enter the ocean is that it undergoes degradation. And there's 5 main methods of degradation that plastic undergoes in the ocean. One is photodegradation, which happens by UV light or other light exposure. thermo-oxidative degradation, which is the gradual oxidative breakdown of the plastic in moderate temperatures. Thermal degradation happens when the temperature of the water is high. Hydrolysis, which is the reaction with water. And finally, biodegradation, which happens when the plastic reacts with different microbes. And in my study, I look at microbes that are potentially capable of causing biodegradation. And this graph basically shows the different number of years various types of plastic items take to biodegrade in the ocean. And these are just estimates. However, there's a large variation between the different items. And this comes down to before when I said it's hard to generalize different plastic items as one thing, as they're made up of different polymers and additives, which has resulted in this variation in terms of the number of years they take to biodegrade. However, biodegradation is caused by marine microbes, as I mentioned before, and they come from all three domains of life: bacteria and archaea, which are prokaryotes, as well as eukarya. If you take 1 milliliter of seawater from the ocean, that you will find around 10 million marine microbes living in that 1 milliliter. And that is how abundant marine microbes are. So naturally, if you discard your plastic items in the ocean, they are going to get colonized by marine microbes. And the marine microbes that colonize marine plastics in the ocean are found to be unique and different to the surrounding seawater and the microbes found in the surrounding seawater. And because of this uniqueness, this community living on plastic is now referred to as the Plastisphere, which is the topic of my study. So now I will show you a few pictures of from a scanning electron microscope, a few time series of a HDPE water bottle, and this is time 0. And you can see how the plastisphere develops with time. And there is a clear difference between time 0 and 16 weeks. You can see different microbes colonizing the plastic. And here you can see some diatoms clearly in week 1. It is, however, degradation is not too clear in these pictures. Therefore, I've included a different picture from a different paper where you can clearly see the pits that had conformed to the bacteria, and where you can see the degradation clearly happening caused by the Plastisphere species. However, coming back to that original study, this graph basically shows the taxonomic classification of the different microbes that were present on this 1 gallon water bottle. And what I want to explain is that the Plastisphere is not comprised of one microbial species. As you can see here, it varies with time, and it doesn't remain consistent or constant, sorry. And this map basically shows the different studies, Plastisphere studies, with the use of high throughput DNA sequencing that had been happening around the world. And it is pretty obvious that this is as of January 2020. And Australia has not been up to speed with the rest of the world in terms of Plastisphere studies, which is what I'm trying to tackle. And this is also important to mention. After my lit review, I noticed that the average time of a Plastisphere study is very short. And the longest I could find was this paper that went on for 12 months. They looked at how the plastic surface changes with time, but however, they didn't address the microbes that were living on it. However, after my lit review, I've come across some unanswered questions. How these different types of plastic may change the Plastisphere. As I mentioned, that plastic is not one thing. They're comprised of different polymers and additives, and we don't know how these different polymers and additives may change the plastisphere composition. Secondly, how the plastisphere composition changes with time. As I mentioned before, the plastisphere studies are not too long, the ones I've come across. So we don't know what happens after those average 8-week period. Does the plastisphere keep changing forever, or does it reach a plateau? And how environmental changes can impact the plastisphere. So it's obvious that plastic is going to be a part of our oceans for the next decades and centuries, and it is also obvious that that we've come across these climate change, oceanic changes such as climate change, and we don't know how these climate change, such as sea temperature, sea surface temperatures changing, may impact the plastisphere in the marine plastic. Also, I'll come back to this a bit later, but there's also evidence in other parts of the world where plastic in the ocean has acted as vectors to opportunistic pathogenic marine microbes. And it is also known to influence metal and chemical pollutant accumulation. And finally, there's ample evidence that different marine species engulf plastic, and the presence of a plastisphere on these plastic, how does this impact these marine species? That is also unknown. So to try and answer these questions, I've developed these my objectives of my PhD. And firstly, I will identify the plastisphere species through a 12-month time period to see the variations see the composition changing. I will investigate how sea warming may impact the early colonization rates of the plastisphere. And finally, I will look at plastic samples from the coral sea region, and look at different microbes and potentially pathogenic microbes that may be hitchhiking rides on these plastic in these areas. So firstly, my first objective is a long-term study to identify the different plastisphere species using high throughput sequencing throughout a long time period. And I want to observe the temporal succession, so I will be deploying plastic samples for a 12-month period from start to end. And in the first month, I will be taking out plastic samples every week, followed by monthly, until 12 months to see how the composition has changed. And here it is also important to mention that I'm incorporating all the main 6 types of plastic in this objective. And the study site I have chosen for this needs to be easily accessible, as I will have to visit it and take out samples very often. And also it needs to be big enough to hold multiple samples. And for this, we've chosen a marina berth. And we understand that the data produced within a berth is not generalizable to the rest of the ocean. So we have collaborated with Ocean as well as the EPA, who have mooring stations such as these, to put out plastic samples in the open oceans. These are some of the locations that Ocean have mooring stations, and these are some of the locations I would be looking at the plastisphere in. And also I will be using high throughput sequencing, more specifically the 16S ribosomal RNA gene, and I will be focusing on the V4 region of this gene to identify the microbes. And I'll also use the scanning electron microscope to generate images such as this. Secondly, I will also include a Mesocosm Lab experiment where I will basically replicate current sea surface temperatures. So these are the sea surface temperatures of Birkenhead, which is close to my marina berth. And I will replicate these temperatures in the lab with plastic incorporated in the tanks. And I'll also have 2 other treatments with 1 degree and 3 degrees warmer to the current sea surface temperature, and see how the plastisphere adapts or changes to these warmer conditions. So for this, I will not be doing it for 12, I'm not planning on doing it for 12 months. I will look at the early colonization only, so the first 4 weeks. And here again, I will be incorporating all main 6 types of plastics to see if there's any difference within the different plastic types in terms of the plastisphere in warmer waters. And finally, I will be taking out samples from the Coral Sea region, wild plastics, and I will be looking at, similarly to my first objective, I'll be looking at the different plastisphere species that are found on these plastic. And here I will be paying more attention on pathogenic microbes that can be potentially traveling on plastics in these areas. And there's evidence to this, such as Vibrio species and Pseudomonas. These 2 pathogens have been found to have traveled on plastic pieces in other parts of the world. And these species, the Vibrio species is capable of causing diseases such as vibriosis, as you can see here. Another pathogen was the ciliate that was found on plastic in another part of the world. And these, I think it was the Northwestern Pacific, however, these pathogens are capable of causing a disease called the skeletal eroding band disease in corals, which looks like this. And this is also one of the main reasons why I want to keep a key importance on these pathogens in this objective, because in this Coral Sea region, we've got ample coral diversity that may be impacted by such microbes that may be traveling on plastic. So, which is why I'm focusing on pathogens mainly in this objective. So, however, to get these samples, I have collaborated with X-Expedition, which is a sailing voyage that's traveling around the world, trying to tackle and help oceanic plastic research. And they have agreed to provide me collect samples from this region, and where I can observe how the plastisphere is looking like in these areas, as well as looking out for different pathogens that may be found on the plastic. Finally, the contributions of this project will be, I will be identifying the key plastisphere species, and I will be observing biodegradation, because my study, the first objective will be a long-term study, as well as I'll be incorporating all the main types of plastic to see if there's any difference between the 2. This can be possibly used for any biotech innovations where we can harvest microbes from the ocean in order to recycle plastic, which is a big problem right now, as we all are aware of. Secondly, I will also pay attention to the pathogens that may be transporting across our oceans on plastic, and I will look at the focus on human pathogens and pathogens that may impact marine species that may be impacted because of the plastic that has these pathogens living on them. And finally, I will also look at pathogens that can disrupt ecosystems, such as the ciliate I referred to before, that can spread diseases among corals, which are the base of huge ecosystems. Finally, I will also look at how sea warming impacts the plastisphere, which I have not come across in other studies so far. And we will be able to predict how the future seas, oceans will be looking like in terms of the plastisphere and plastic. And there's also a chance to collaborate with Sophie, who is presenting after me, to see how metal and chemical pollutant accumulation may be impacted by the plastisphere species. And that is all I have today. Thank you for listening, and it's over to you, Bobby.
Awesome. Thank you, Vinuri. That's such a awesome project you've got there. I love how it's bringing in a range of collaborators, people and other organizations, as well as research methodologies there to, address such an interesting and important issue in our oceans. So we'll move on now. Our next speaker today, as I said, another PhD candidate in the start of her journey, Sophie, and she's bringing together a range of disciplines as well, bringing the chemistry to the marine biology as well. So, Sophie, I'll hand over to you and let you talk.
Beautiful, thank you, Bowie. Just get my screen going. All right. Cool. So as Bowie said, my name is Sophie Dolling, and I'm going to be telling you about my PhD project. So I'm investigating plastics as a vector for chemical pollution in seafood species, and I'm supervised by Professor Bronwyn Gillanders and Dr. Mike Williams from CSIRO Land and Water. So first of all, I'm going to tell you a little bit about plastics. So Vinuri has given you a good understanding about plastics and plastic pollution, but I'm going to take you through the chemistry behind plastic pollution and its effects. So firstly, plastics are organic polymers that are synthetically created for human use. So they're composed of reoccurring simple organic molecules, and they're petrochemical based, so they're derived from oil. So they've become more popular since the end of World War II in the 1940s, and that's because they're incredibly long lasting and they have a range of different polymer types that can fit huge amounts of applications in society. So they're really useful things. So some of the most common polymer types include polyethylene. So polyethylene is things like your plastic bags, cling wrap, and chip packets. So that's like the soft type of plastics that are very malleable. Polypropylene. These are things like plastic bags, cling wrap, or straws, cutlery, and biscuit trays. So we've also got polystyrene. So this is things like your foam takeaway containers, coffee cups, and packing materials. So things like styrofoam. We've also got polyethylene terephthalate, or PET. So these are plastic bottles. And here in South Australia, we are lucky that we're actually able to recycle these and get a 10-cent refund on our PET plastic bottles. We've also got polyvinyl chloride, or PVC. So this is an industrial type of plastic, commonly can be known as polypipe. It can also be used in wall cladding, electrical plastic, and those kind of industrial type applications. And lastly, this is one that people don't often think about, but polyester. So polyester is normally used in clothing, as well as ropes and fishing gear. So when we actually wash our clothes in the washing machine, thousands of little fibers actually come off of our clothes and can be washed into wastewater. So this can actually be a really big input of plastic pollution into the marine environment. And it's something that I didn't really think about before I started studying microplastic pollution. So if we go back in time to 1950, the world's population stood at roughly 2.5 billion people, and the amount of plastic produced was roughly 1.5 million tons. So if we fast forward to 2016, which was a few years ago now, we had a global population of 7 billion people, and we produced 320 million tons of plastic. So you can see that there's been an exponential increase since we started using plastics in the 1950s. So if this growth continues at this rate, we can see this number of 320 million tons, we can see this double again by the year 2034. So you can see that the use of plastic is really on the rise. So the amount of plastic being produced and polluting the environment is likely to see it become the geological indicator for the Anthropocene era. So we know that plastic pollution is ending up in the marine environment, but is this actually having an effect on our marine life? So there's been a lot of research into the effects it can have on marine life, and I'm going to go through some of those examples now. So, for example, marine megafauna like your bigger whales and sharks and those kind of things can become entangled in plastic pollutants. So on the screen here, you can see an example of a whale getting entangled in discarded fishing gear. So this is a really big problem for those bigger organisms that need to continue moving to breathe through the water, so it can have detrimental effects. Also, many marine organisms can falsely identify plastics as a food source. So on the screen here, we've got a turtle going after a plastic bag. So to a turtle, a plastic bag and a jellyfish, which is one of its main sources of food, are going to look very similar in the water column. So this can also happen for fish as well. They're going to see a brightly coloured little piece of plastic in sediment and think it might be a really nutrient-rich piece of algae or something like that, and they're going to misidentify that plastic as a food source. So this can lead to something called false satiation of the organism. So basically what that means is the organism becomes full of plastics, and because its stomach is feeling full, it's satisfied. So they're not going to go after any other food sources, but the plastic that's in its stomach doesn't have any of the nutrients that they need to sustain life. So essentially they're starving themselves from the inside out because they're not getting any nutrients from what's inside their stomachs. And lastly, the ingestion of plastics can cause choking, perspiration of internal organs of the organism, and can have some really detrimental effects. So I've highlighted some of the physical effects of plastic pollution, and these are some of the things that we see in the media. But when organisms are ingesting plastics, there can also be some really bad effects on the chemical level, but this hasn't been researched very widely. So in the plastic production process, a number of chemical additives are added to plastics to make them fit a specific purpose. So this could be to make the plastic more rigid, it could be to make it more malleable, flame retardant, or antimicrobial for food plastics. So there's a range of different reasons that chemical additives can be added to these plastics. Now these additives, they're not actually chemically bound to the structure of plastics. So when the plastic begins to degrade in the water column, they're released quite easily. So this could include compounds like BPA. So this was a common additive in food plastics, but it's under a lot of regulatory concern now. Could be something like 4-nonylphenol. It could be polybrominated diphenyl ether or PBDE, or it could be things like polychlorinated biphenyls or PCB. Now, these are just a few examples. There's thousands of different additives that are added to plastics, but these are just a couple that I thought I would show you. So when these additives are released, they are commonly hydrophobic. So this means they're water hating, similar to if you were to put oil in water. So basically they hate the water, they want to get away from the water as quickly as they can. So this means they'll absorb to anything else they can in the water column. Now, as they're being released from the plastics, most often, this is actually the microplastic surface itself. So when a marine organism consumes the plastic pollution, it will not only be consuming the plastic, but also a suite of chemical pollutants that have absorbed to the plastic surface. So previous studies have found that hydrophobic contaminants can be found in concentrations of up to 500 times higher on the plastic surface than what these contaminants are found in the water column. So you can see there's a lot of accumulation happening on the surface of these plastics. So there's evidence that suggests that both the microplastics and the chemical pollutants brought with them are able to translocate into the tissues of marine organisms. So these pollutants can then be transferred to high trophic level species, and this could cause bioaccumulation through the food chain. So I've talked about chemicals that come from plastics, but can any other chemicals act in this way? And the answer is yes. So it's been theorized that other chemical pollutants like pharmaceuticals could possibly act in this way. So you can see on the screen there, we've got diclofenac. This is commonly known as Voltaren. So a lot of you have probably used Voltaren before. So this could possibly act in this way. Pesticides from runoff, such as atrazine, could act in this way, as well as heavy metal pollutants from industry. So why is this a problem? Why should we care if these chemical pollutants are ending up in fish? Well, many of the pollutants we've discussed can have detrimental effects on both marine organisms and the humans consuming them. So for example, we talked about BPA earlier. BPA is known to be an endocrine disruptor. So essentially what that means is that it can affect hormone systems and negatively impact the reproduction systems of the organism. Also, BPA has been linked to cancer, so it could be carcinogenic, and also linked to heart defects. So there's some pretty bad things that can happen through inducing BPA. So this brings me to the gaps in knowledge for this theory. So it's still unknown as to what chemical pollutants can absorb to the plastic surfaces in the water column and at what concentrations this is happening. So a lot of the things I talked about today are mostly theorized. We haven't actually tested them in a real setting. And as Vinuri mentioned earlier, concentrations of some pollutants on plastics could be affected by the biofilm present, but we don't actually know these things either. So it's still not known what concentrations of chemical pollutants are being translocated to marine species. And this is largely because appropriate analytical methods for analysing pollutants in fish tissues haven't been developed, and then they haven't been standardised. So the development of a method for a low concentration analyte in a complex matrix like a fish tissue that can analyse multiple analytes, being the chemical pollutants of interest, at once is really needed, so we can gain more of a grasp on the spread of chemical pollutants in marine organisms and really study this interaction between the plastics and the chemical pollutants in the water column. So this brings me to my project. So I'm going to be investigating the ways key contaminants are able to absorb to plastic surfaces and then be transferred to marine organisms. So I've got a few research aims. The first one is to investigate a method for pharmaceutical pollutant analysis in fish tissues. Secondly, I'd like to identify the presence of chemical pollutants in common seafood items across Australia. Thirdly, I'd like to investigate the interaction between the priority chemical pollutants that are found in the water column with the plastics in marine ecosystems. And lastly, I'd like to investigate the residency time of plastics and chemical pollutants actually in these marine species, so in the gut and the tissues. So let's talk about number one first. So this is to investigate a method for pharmaceutical pollutant analysis in fish tissues. So in recent history, a lot more attention has been paid to the presence of pharmaceutical pollutants in our oceans. So pharmaceuticals can enter the environment through wastewater or through incorrect disposal and are thought to be essentially medicating the environment. So what this means is that pharmaceutical pollutants could be absorbed by marine organisms and unintentionally medicating them. So as I highlighted earlier, pharmaceuticals could have the potential to accumulate on plastic pollution, and they could cause high concentrations on this plastic surface being transferred to marine organisms when they're ingesting the plastic pieces. So obviously this could have detrimental effects on the organism. So it could affect their feeding, it could affect their breeding, and therefore affect their population growth. So to better understand the effects of pharmaceutical pollution on fish, we need to come up with an analytical method to identify the presence of this pharmaceutical pollution in the fish tissue. So for this project, I'm going to be looking at a range of different pharmaceutical pollutants. These were chosen based on their use in Australia, as well as their identification as pollutants of concern internationally. So a multi-residue extraction method will be developed using accelerated solvent extraction, or most commonly known as ACE. So essentially, this is how we're going to extract the pollutants from the fish tissues themselves. So how this works is by pumping a specific solvent into the sample under pressure and heat. The solvent will then be extracted through the sample, bringing with it the analyte of interest. So basically the analyte will be dissolved in the solvent. So ACE is a really efficient technique because it's fully automated. So the process takes approximately 30 minutes per sample, and around about 18 samples can be analysed per run. So it's like a set and forget type of analytical technique. So we can put it on and then come back a few hours later and they've been looked at. So I'm going to be testing a range of different solvents as well as operational parameters like heat and pressure to ensure the greatest proportion of pollutants can be extracted from the fish tissues. So once we've got the extracts out of the fish tissue, we then need to come up with some way of actually analysing them and seeing what's there. So this is done through an analytical method, either by gas chromatography mass spectrometry, more commonly known as GCMS, or liquid chromatography mass spectrometry, or LCMS. And it will be depending on the physicochemical properties of the compounds being analysed as to which analytical method I use to look at them. So again, the aim is to create a multi-residue method to ensure there's an efficiency of analysis. So this will really become helpful in later chapters when I'm looking at larger sample sizes and a huge range of different compounds. So by looking at multiple residues at once, I can get through this process quicker. So onto my second aim, which was to identify the presence of chemical pollutants in common seafood items across Australia. So fish samples were collected from all of the coastal capital cities in Australia by Nina Wooden, another PhD student, and this happened in 2019. So from these fish samples, she actually collected tissue samples from each specimen. So this included both liver tissue and muscle tissue. Now these fish samples were sourced from markets, and we chose to do this to best represent how a consumer would be sourcing their fish. So some of the species that we'll be focused on for this chapter include the Australian salmon, garfish, King George whiting, flathead, Tommy Ruff, Australian snapper, blue mussels, and the Pacific oyster as well. So all of these species are of high commercial importance to the Australian seafood industry. So understanding the chemical pollution that might be in them is really important. So from each individual fish, like I said before, the liver and the muscle tissue was collected. So these tissue samples will then be analysed for the presence of a range of chemical pollutants. So some of these pollutants could include diclofenac. So this is from the pharmaceutical kind of class. We're going to be looking at endocrine disrupting agents as well, things like BPA that I talked about before. I'm also hoping to look at polybrominated diphenyl ethers, organochlorine pesticides such as atrazine, and polychlorinated biphenyls. Now, these last three that I mentioned are all of high regulatory concern, so they're considered to be pollutants of concern internationally. So that's why I think it's really important to look at them in Australia. And I'm also hoping to look at heavy metal pollutants from industry such as mercury and lead. So these compounds will be extracted by accelerated solvent extraction, like I spoke about before, and will also be analysed by either GCMS or LCMS, depending on their physicochemical properties. Now, onto my third chapter, which is to investigate the interaction between priority chemical pollutants and plastics in marine ecosystems. So essentially, this is the interaction that I'm going to be focusing on. So I'm trying to figure out what pollutants from plastics are absorbing to the surface of microplastics in the water column, but also what outside pollutants are acting in this same way. So when I say outside pollutants, I mean pollutants that have not come from the plastics themselves. And it's been theorized that a range of pollutants are able to absorb to the surface of the microplastics. But like I said before, this theory is really under-researched, especially in a field setting. So to test this interaction in a real marine setting, I'm going to be deploying plastics from a berth at the Cruising Yacht Club of South Australia in North Haven, so the same as where Vinuri is going. So I'm going to be analysing 5 different plastic types that have been shredded to the micro size, and this size will be checked using a light microscope. And as I'll be sourcing the plastics from societal examples such as bags and bottles, I'm going to be checking the polymer type of these plastics also using FTIR chemical analysis. So these plastics will then be submerged in the water column in mesh bags with a mesh size of approximately 100 micrometers so that all the plastics will stay inside those bags. So this is kind of what it's going to look like. Bags will be attached to weights to ensure they stay submerged under the water column, and then attached to a central line across the berth. Now floats will be on this line so that the bags will move throughout the water column with the change in the tide. So I'm going to be sampling plastics over a 12-month period. So sampling will be really heavy in the first 3 months, and they will become less frequent in the remaining 9 months. So this sampling regime was set up because each plastic has a different equilibrium point. Now, the equilibrium point is the time where the chemical pollution on the plastic surface won't increase any further, so it will simply plateau. For some plastic types, this can occur in the first 3 months of submersion, but for others, it can take up to 12 months. Most previous studies have found that the equilibrium point occurs mostly in the first 3 months, but there are outliers based on the plastic type or the environmental conditions. So by taking a snapshot of 12 months, hopefully I'll find the equilibrium point of each plastic type. So once the plastics are sampled, the same chemical analysis described earlier will occur on each plastic. So firstly, I'm going to extract the chemical pollutants from the surface, followed by analysis using either GCMS or LCMS. And lastly, my last aim was to investigate the residency time of plastics and chemical pollutants in the gut and tissues of marine species. So this is going to be a captivity experiment, which will be undertaken using either blue mussels or oysters. So this experiment will involve the sedentary filter feeding organisms being fed a plastic feed mix. So the plastics will be taken from the previous experiment, having accumulated chemical pollutants in a marine setting over time, and then they'll be fed to the sedentary filter feeding organism. So the mussels or oysters will be sampled at different time periods over a 3-month period. And at each sampling time, they'll be analysed for both their plastic accumulation, so that's actually individual pieces of plastic, as well as tested for chemical pollution using methods outlined previously, so the ACE and the GCMS and LCMS. So by sampling at different time periods, it's hoped that we'll be able to deduce the residency time of plastics and other pollutants in the organism. So the expected outcomes of this research is to better understand the interaction between plastics and chemical pollutants and how their effects on seafood can possibly lead to potential changes in the way society uses and disposes of their plastics. I'm also hoping that understanding this interaction more will allow more informed decisions to be made by policymakers in SA and nationally, both on our use of plastics and our seafood policies as well. I also hope to create a standardised method for testing chemical pollution in fish species, and I'm hoping this will lead to better understanding the chemical pollution currently present in the Australian seafood industry, and by having a standardised method, we'll be able to place this in an international context. And that is everything. Thank you for listening.
Awesome. Thank you, Sophie. Another great presentation. And yeah, thank you to both of you for joining us again to get this recorded. I'll just finish up by saying that this is our monthly seminar run by the University of Adelaide, and we do this on the first Friday of every month. And so next month we have 2 speakers. Jenny Weinheimer is a master's student at the start of her master's, looking at conservation's money problem, as well as Pam Catchside looking at the fungal diversity and that amazing community that we know so little about. So join us next month on the 6th of November for that one live, or watch the recordings here. Thank you for joining us, and thank you to our speakers again.